There's a lot to be told about our own research past on heart disease, the connection between heart disease and dental diseases. These are the topics that we're going to be talking about tonight. I owe you a little bit background of what we have been doing, me meaning myself and the research team that works with me. Micronutrient deficiency, oops, there we go. So, micronutrient deficiency in dental diseases and late research in dental health that, to our understanding, to our knowledge, is unique in the world and may actually contribute to improve the preventive approach to dental health, not just in the Netherlands, but way beyond. My main interest through my professional life was on cardiovascular disease. Until this day, every third person on this planet dies from this disease, men and women alike. There are major problems, unanswered questions that made me pick up this direction. One of them was, why do we get infarctions in the heart and not in the nose or elbow? If cholesterol damages the arteries, it is the same level in all arteries, veins, capillaries. Why do we get arteriosclerosis and not venosclerosis? Again, the cholesterol levels are the same in arteries and veins. Why do people die in epidemic proportions from a disease that is essentially unknown in the animal world? And there's a law in medicine. An epidemic can only prevail as long as the true cause of the disease is not understood. It's a logical thought, but it is important to remember for therapists and patients alike. This molecule here changed my life. Without that, I wouldn't be standing here. For about half a century before I went into research, medicine looked at this little molecule, this little molecule here as the main culprit. It is low-density lipoprotein, a cholesterol fat transporting particle that swims in our bloodstream in this form as a spherical particle. The reason why they thought this is the culprit that causes the death of heart attacks and strokes was because they looked into human arteries from operations or after people died, and they identified this molecule in masses. So they said, well, if we lower this molecule, i.e. cholesterol or statins, will help people. We found out at the university research in Hamburg 30 years ago that in order for this particle to stick in your blood vessel wall, it needs an adhesive tape, a biological adhesive tape, a very sticky protein called apoprotein A. And together, this spherical lipid cholesterol transporting particle and this adhesive tape, they unite and they provide a new molecule called lipoprotein A. And that is what causes heart attacks and strokes, not this. The reason being significant. Statins do not affect the blood levels of this molecule. They do this, but not this. Today, it is clear that lipoprotein A, or short LP little a, and not LDL, is the main risk factor for coronary artery disease, cerebrovascular disease that causes strokes, for peripheral vascular disease, when you have clogging of the arteries in the legs, for example, restenosis after bypass surgery, when the arteries clog after the operation, and calcification of heart valves. This is the discovery that I shared in 1988 with leading researchers, including Nobel Prize winners. What does it say? There was an interesting observation I made, and that was this risk factor, lipoprotein, that kills literally millions of people, is found in humans and subhuman primates, but not in the rest of the animal world. And interestingly, there is an inverse relationship to the production of vitamin C in the body. Most animals can produce vitamin C, but they do not produce this risk factor. Vice versa, we humans lost the ability for endogenous, meaning vitamin C production in our body, and instead we have this very dangerous molecule. What is this about, this relationship? Now, most of the scientists that I talked at the time, they said, "You're crazy. It's just outright crazy. Forget it. Help us sell statins. That's your job." There was only one person that listened to me, and that was Linus Pauling. And he, of course, had worked on vitamin C, and he thought this is not just an observation, this is a principle of nature. And he invited me to join him in California, which I did. And together we wrote several papers with this picture in their centers. Let me explain. Animals produce their own vitamin C. Vitamin C is the single most important molecule for the stability of the body by producing the collagen molecules, thereby stabilizing the artery walls, and for that matter, teeth too, as we will see later. Humans, we get little vitamin C in our diet. We do not produce it in our body. So there's a constant problem of stability in our body. And suddenly we understand why we humans, our ancestors for that matter, had a great need for molecules that help them to do what? Repair, in particular, an increasingly destabilized blood vessel wall. Because if there's no repair, what happens then? Well, you are from a seafaring nation. You know what the people 400 or 500 years ago died of that dared to cross the ocean sea. They died of scurvy. So the inability to produce vitamin C in the body, in the human body, in combination with too little vitamin C intake in the diet, causes death. And suddenly we understand why long ago, centuries, millennia ago, only those people survived that had repair molecules to make them survive. And only they could have children. And you all, including myself, are the offsprings of those who had inherited this molecule. Am I losing you? We wrote several papers together, Linus Pauling and I, in 1990, 91, 92. The title of these papers, the titles, could not be clearer. Solution to the Puzzle of Human Cardiovascular Disease. A unified theory of human cardiovascular disease leading the way to the abolition of this disease as a cause of human mortality. So there was no mistaking on what we thought we had discovered. Did we make friends in the pharmaceutical industry with these publications? You bet we didn't. The last public appeal that two-time Nobel laureate Linus Pauling wrote and signed in his own handwriting stated, the goal of eliminating heart disease as the major cause of death and disability is now in sight. Almost 30 years ago. Linus died in 1994, and he handed over the torch to me. He said, you got to do this. There's no one else in my institute. I said, Linus, there's no one else in the world. And he had tears in his eyes because he knew how hard this battle would be. We confirmed everything that we've published as a concept at that time. In the meantime, scientifically, these are humanized animals, mice. I know about animal experiments. We are not taking that easy, but some questions can only be answered in selected experiments. And so we turned mice into humans as it relates to this risk factor. And these mice that we used could produce human lipoprotein A. And we turned off the genes for vitamin C production. So we made them humans in two critical aspects. No vitamin C production in their body, but human lipoprotein A production. And of course, we wanted to see what happens if these animals get too little vitamin C in the diet. We wanted to mimic the human situation. And here's the result. These are both groups of animals did not produce vitamin C in their diet. These are the animals that, in addition, did not get enough vitamin C to keep the artery walls stable. And what you see here, these plaques, they develop in these animals. Both animal groups had the same levels of cholesterol and fat in their diets. So there's no cholesterol feeding involved in this plaque development. And of course, these deposits contained lipoprotein A and made us understand what that particle really is. It is a repair factor that the body needed to protect our ancestors from dying. And if this repair process overshoots, causes atherosclerotic plaques in millions of people. We published this in the American Journal of Cardiovascular Disease in 2015. Again, you don't need to write this down because you can get it from the video later. Clinical confirmation. We did a clinical study to prove this. And this is the ultra-fast computed tomography where you can measure calcification in the coronary arteries of patients. Now this is, you look from the bottom into the heart of the patient in this technology. What you see here is the left coronary artery. The white spot here is the calcification in the left coronary artery. 1 year later on a vitamin program, same patient, same position in the heart, same left coronary artery, no more calcifications. This was possible in beginning calcifications. In advanced calcifications, we were able to prove that you can stop the further development. But the complete disappearance would have been taking longer on that. But this was the first time in the history of medicine that a non-invasive way of a non-technical way, non-operative way, bypass operation or balloon catheter, showed the disappearance of calcification in human coronary arteries. And suddenly we can understand and answer those questions that none of the cardiologists at the University of Utrecht, Amsterdam, New York, Vladivostok can answer today. Why do we get infarctions of the heart? Well, very clear. The heart is the only organ in the body that does what? Moves, pumps. The coronary artery is right on top, and with every heartbeat they are squeezed flat. So there, where the mechanical stress is where the underlying instability of the blood vessel wall is exposed first. And this is where the plaques, the repair, overshooting repair develops. And we understand why we get atherosclerosis, not venous sclerosis, because there's blood pressure about 80 times higher in the arteries than in the veins. Again, that is mechanical stress on the walls. And we understand, of course, why animals do not get heart attacks, but people do, because they produce their own vitamin C and they don't care about cholesterol. Bears that go into hibernation have a cholesterol level of 600 to 800 milligrams per deciliter. They wake up happily next spring. They're not brought to the intensive care unit or put on statins. If you want to know more about this, I have only limited time to talk about this, obviously. So outside is a booth. You can get this book where the things that I touched on and I couldn't touch on are described in a understandable language, because from the very beginning we were convinced that change will only come by the participation of the people. Only if we reach and talk the language of the people will we be able to make change. And I guess the composition of this audience reflects that too. At the end of this book, you will also have a chapter on the battle that started immediately when we came out and how we were able to change the laws in the United States, establish a law that the truth could be said about micronutrient research, something that we are still waiting for in Europe. Well, I see in your faces that some of you look at me in disbelief and say, well, if you were right, I would have heard about this. Well, maybe, maybe not. The forces that surround the pharmaceutical investment business are quite strong. They not only control politics, they also control the media. Wikipedia has a foundation, and the Wikipedia Foundation is controlled by Mr. Soros. Mr. Soros is one of the greatest investors in the pharmaceutical business. So anything you read about Dr. Rath on Wikipedia, you know where it comes from. I heard that there were some Facebook comments about me standing here. Where do these come from? So while you may not have heard about any of this, things have happened nevertheless. This is the explosion of scientific literature on the health benefits of vitamins. From 1990, when we published these papers, to today, more than 60,000, maybe now it's over 70,000. In parallel, this is the global production of vitamin C in tons. It was 3,000 before we published. Within 5 years after we published, it doubled to 6,000. Today, it is almost 100,000. So the news spread around the world. Even so, no one talks about Dr. Rath on Dutch national television. It's a reality nevertheless. And I'm happy to be here to talk to you about this today. But that's not all. These are statistics, global statistics from 2017. That is the global percentage of people living with vitamin C deficiency. In the years from 1990 to now, actually to 2010 already, the number of people, we're talking about hundreds of millions, who lived in vitamin C deficiency condition on the border of dying from scurvy were cut in half. And this is the latest statistics of countries that in the same time span, the last 20 years, reduced cardiovascular mortality, including the Netherlands. So now I come to the second part, why this information matters to people with dental diseases. These are statistics from Lancet from 2018. Very conservative, as you know if you're in this field. 24% of the world's population suffer from caries. Some statistics say half of the population in the US suffers from caries. Periodontal disease, the instability of the teeth, 10%. Whatever the percentage is, it means it's a global epidemic. And as we already knew from cardiovascular disease, any epidemic that cannot be controlled means that we do not really understand the cause of this epidemic. I've talked about scurvy on heart disease. Let's talk about scurvy in dental disease. I came across a publication that I want to share with you. There's an Indian farmer coming to a clinic in India and reporting about teeth problem. We will see in a moment what it is. And because he had the problem eating of the pain from the gums, inflamed gums, he stopped consuming his regular diet and was only on liquids for the past 15 days. So there was this vicious cycle starting in his life. His teeth prevented him from being able to get nutritious food, micronutrients, fresh fruits, etc. The examination showed that there was spontaneous bleeding on slightest provocation. This is a picture of his teeth. One, there are several more. Well, let me point out, you've seen some of the picture in the previous talk. Look here, swollen gums. Microscopic investigation showed that there was bleeding, blood vessel rupture. Sounds familiar? Blood vessel rupture in the tissue that you just saw in the mouth when it was removed. This is an X-ray. You see the same thing you see in periodontal disease, that even the bone is decayed. Then they did immune function tests on white blood cells, and they found that there's a decreased ability to reach the invading bacteria. It's called reduced chemotaxis. There's a decreased ability to kill the bacteria. There's reduced phagocytosis. And then they gave this, of course they did the standard procedure to clean up, and then they gave this man 1 gram of vitamin C per day, and these are the pictures after 3 months. Again, this is the PubMed code to get access to this study, but you can get it from the video. I think anyone interested in preventive dental health should check out this study. So what is the scurvy cardiovascular dental disease connection in detail? Well, we can summarize it already. The common cause, the primary cause for both diseases is vitamin deficiency, lasting not just for a week, but for years, for decades. If you think that the recommended daily allowance of 70 milligrams, or I don't know what it is currently, maybe it's already down to 60 milligrams, is enough, you have to think twice. Animals produce, compared to the human body weight, between 10,000 and 20,000 milligrams each day. We already know that vitamin C is responsible for producing collagen and creating the stability. Now, if you have too little vitamin C, you have a deficiency in collagen molecules, destabilize blood vessel walls, the gum bed, bones, all are destabilized. All tissue is destabilized. Heart disease, dental disease. Common cause. Through an understanding of the molecular basis of diseases, not because I'm saying it. If we look at the dental problem in more detail, we heard about caries, we heard about periodontal disease. The difference, as most of you know, but just for the patients, is that caries is more superficial here. Periodontal disease is the name for at the side of the tooth disease, where you have a gap, where bacteria are settling, where you have inflammation, and where you have decay even of the bones. Now, the therapists among you, pardon me that I'm talking about this, but it's important to briefly reflect on this. Here's a picture of what is called dental plaque under the microscope. These are bacteria that build their own nest. That's plaque. They like to be there for a long time. So they build their nests. And they secrete material, long sugar molecules, proteins, in which they settle. In short, and again, I cannot talk about the details of all these different mechanisms, how they are being blocked by micronutrients, but just as a summary, science today knows that all these mechanisms, starting from the plaque formation, inflammation, tooth decay, and of course the systemic spread of disease through signal molecules, so-called cytokines, all of them can be blocked by micronutrient compositions, vitamin C being the most important among them. By the way, if there is an interest among you, patients or therapists alike, to know more about all these things, we are able to provide this information in lectures, etc. So if you're interested, you can sign up outside on the list, and Terry, is Terry here? We'll be able to coordinate it with you, and then you have the possibility to get more information on these mechanisms. For now, I'd like you to understand another aspect. This is, if you want, an escalation of the dental disease problem. Caries, the beginning, pulpitis, meaning there is an inflammation of the interior of the tooth, gingivitis, the disease spreads to the outside, and there's inflammation in the apparatus that holds the teeth. And then periodontal diseases, where you have a gap here between the tooth and the the apparatus that holds the tooth in its position, including the bones. So, what do we observe? We observe that at the beginning of the disease, there is a little inflammation involved in the surrounding. Then we see that as the diseases progress, more and more cells are being involved in this process, and they are not healthy cells. They are activated cells that are, we call, a disease. And what do these cells do? They produce factors that stimulate the formation of new blood vessels. They favor infection. They favor biofilm formation, etc. The question is, was there a benefit for dental diseases? Was there a benefit for our ancestors to be susceptible to developing caries and developing periodontal diseases? I know it's a provocative question, but here is a possible answer. I was invited to give a thought-stimulating talk, so there you have it. Imagine our ancestors living in times of drought or ice ages or other challenges where micronutrient deficiency was the rule. They had two options. Either they die from scurvy, or they come up with mechanisms under micronutrient deficiency that cause a repair system to be activated. Now, in vitamin deficiency, you already have the opening of the gaps aside from the teeth, because we all know that the sailors died, or the first sign of scurvy was bleeding teeth, bleeding gums, sorry. So there was a process going on there. But you needed to have the systemic distribution of these signal factors in order to give the answer to this problem. So only if you have from these activated cells the distribution in the blood vessel system can you expect that there is a repair system in the arteries that otherwise cause you death, like the coronary arteries or the aorta or other highly endangered arteries. So this picture may also explain something else. Have you seen a dog or a cat that has a prosthesis? Or is periodontal disease a problem in the cattle or in any house animal? It occurs, but it's not an epidemic. So we have to answer those questions if we want to understand diseases. Where do they come from? Did they have a benefit some time ago? Have we not seen this connection? And of course, the most important question deducting from that is, if we understand that they have a common cause, we may apply this knowledge to prevent it, both diseases. This is the last part of my talk. Research from our institute. This is our institute in California. This is Dr. Netzwicki, who's been fighting the battle with me at the scientific level for 30 years now. Why cellular medicine? Well, very simple. Everything that I told you so far has to do with cells. And the cells that are involved in dental diseases are the teeth-forming cells, the fibroblasts, osteoblasts, endothelial cells. And if your body pool of vitamin C or micronutrients is low, you have a precondition for all kinds of diseases to develop, for the very same reason that I explained to you. If it is high, if your patients or if you as a patient take micronutrients sufficient and the right ones, you have the best precondition to prevent these diseases from happening. And the understanding of what cells play, what role they play, and how micronutrients can regulate the DNA of these cells to do something completely different when the micronutrient levels are high. That is cellular medicine. One pilot study that we've done some years ago was the question in periodontal disease, what is the possibility to reduce bleeding? And as you can see from these statistics here, at the beginning of the study, we had 60% of the patients had bleeding on stimulation, and at the end of the 3-month study, on a basic formula, vitamin formula, and specifically developed vitamin C formula, the percentage went down to 9%. If you want to get more information, you can do that online, or outside here is the stand of Cell Expert. They can give you information on the formulas used. New research direction, that is something fascinating, because to the best of my knowledge, this is the first time in the world that data like this are presented. We asked ourselves, what can we do to stop the bacteria that form the biofilm and that are the source of renewed inflammation and renewed propagation of the diseases, whether it's caries or whether it's periodontal diseases? How can we stop them in a natural way? And we took the two of the most frequent bacteria that are involved in dental diseases, Streptococcus mutans and Streptococcus sobrinus. Forget the names, these are just frequent bacteria. And this is perhaps interesting. Bacteria embedded in biofilm are up to 1,000 times more resistant to antibiotics than their planktonic counterparts. So what that tells us is, wow, even if we would take antibiotics, they would have essentially no effect because the nest protects the bacteria in this nest even from antibiotics to a significant extent. So we tested more than 200 natural compounds, as you can see, vitamins, amino acids, polyphenols, etc., to study the question, can we affect these bacteria? Can we prevent the formation of biofilms? Can we possibly even influence existing biofilms? And I will have to go through the next slides with you very quickly because of the time constraint. But you will get the gist of it. You will understand that cellular medicine and natural dental health goes way beyond vitamin C. What I told you about vitamin C was just the path of understanding. Today, our knowledge is that we can reduce the amount of vitamin C that we need to provide health by understanding what other micronutrients work in synergy with vitamin C in order to have a specific desired health effect. So what I'll show you was natural killing of dental bacteria. That was one goal that we wanted to accomplish. Natural killing of bacteria in dental biofilm, something, as I explained, cannot be or can hardly be achieved by antibiotics. Prevention of new biofilm formation. Let's look at the bactericidal effect first, meaning the killing effect of bacteria. And here you see that the combination we developed is able to kill bacteria in a dose-dependent manner. This is 1-to-1, the pure mix. And down to 1 to 32 dissolution, it is highly effective in killing bacteria. Both strains. This is a time-dependent effect, which means you can see that the effect of killing is immediate. These lines show you what happens to this strain of bacteria if you do not have the micronutrients present. This is a picture from the actual tests. You can see here the turbid solutions of the bacteria without micronutrients. And here, when the micronutrients are added, you can literally look through those glasses. Now, the antibiofilm effect. Again, both strains, Streptococcus mutans and sorbinus. Again, we see the dose dependency here, the mix 1-to-1, 1-to-2, 1-to-4, 1-to-8. You can see that you need a little bit higher concentration in order to have this effect. But this is the control. And this is the therapeutic effect. This is a picture that just shows you what, that you can actually measure that, the results from the colorimetric tests that show the effect on the bacteria. This is interesting. This is a tooth model that can be used in these test systems. And here you see the bacteria on these tooth models, controls. Streptococcus mutans, Streptococcus sobranius. And this is the same tooth here that had been treated with the micronutrient formula in Streptococcus mutans and Streptococcus sobranius here. That's important. That is the effect of this micronutrient composition. Again, there's no synthetic composition as part of it, or some hidden antibiotic or chemical involved. It is the understanding of how nature provided us with a solution applied to a scientific setting. And you can see here that even pre-existing biofilm can be affected by these micronutrient compositions. In this case, we studied the killing of the bacteria that settled in pre-existing biofilms. You can see that it takes a little time, obviously, because they are well protected in their nests for this effect to happen. This is also interesting because here we studied the question, is this formula also able to detach existing biofilm, disease-providing biofilm from teeth? In this experimental setting, we needed to have rather high concentrations, but it was possible. So in summary, there now exists a natural, completely natural formula that has been tested in scientific settings. We call it in vitro studies, to be able to kill the most frequent dental bacteria, to kill the bacteria that are embedded in existing dental biofilms, and to eliminate existing biofilm, including the bacteria, of course. Now, I told you I have not come across any of this in the scientific literature. And of course, it would be extremely important to validate these scientific data in a clinical setting. And I would hope that we have the possibility to talk with the organizers, with your organization. Maybe therapists and patients together can make pilot studies beginning next year, and if they are successful, we would have a strong weapon. We can talk about it. If you're interested, Terry is outside, talk to her. That's the conclusion. Together we can reduce the dental disease epidemic. What you can do as a dental therapist: apply micronutrient synergy for treatment of your patients. Systematically introduce science-based natural prevention in your practice. Participate in observational studies, I just talked about it. Spread this health knowledge among your colleagues, and support your patients in their demand to get reimbursement from their insurance for science-based natural dental health. What you can do as a patient: inform yourself further about what you've heard tonight. Start using science-based micronutrient formulas to prevent dental diseases and to support recovery from them. Talk with your family and friends about it, and demand from your health insurance company to start reimbursing for science-based natural dental formulas. I'm at the end of my talk. I thank you for your attention.